Hello, I'm presenting our poster, Structural Characterization of M protein, Insight into a Higher Order Oligomerization in SARS-CoV-2 Envelope and Evolutionary Relationship with WRF3A. This work is done in collaboration between Corking Lab from Worcester Polytechnic Institute, where I'm a PhD student from, and the Martini Group from University of Groningen, Netherlands. Our collaboration started in 2020, where we tried to build an envelope model of SARS-CoV-2. To do that, we need structural information from spike protein, membrane protein, and envelope protein. However, we don't have any experimentally solved M-dimer structure until very recently. So we built an integrative modeling procedure to build our own M-dimer structure. After building our M protein, we observed that it is actually a structure homologue of WRF3A protein, which is another protein of SARS-CoV-2. So we're trying to examine the functional conservation between these two proteins. This is our integrative modeling procedure, where we have the de novo modeling of the transmembrane domain. From our de novo model, we applied a template modeling from the fragments of WRF3A to improve our model. And finally, we run an all-atom simulation to do model refinement and got our final structure. After that, we built a hybrid sequence alignment procedure to compare the functional conservations. We first overlap the WRF3A and M-dimer structure to get our structural alignment. We annotate the charge and polarity, as well as the functional residues of WRF3A protein. And then we combine it with the homologue sequence alignment of each protein, where we finally get our hybrid sequence alignment here. In the results section, I want to first share the structure comparison between M-dimer and WRF3A before and after the structure refinement procedure. We superpose the two structures before and after the refinement procedure, and we observe that the overall RMSD across all residues are very similar before and after the refinement. This is some of the snapshots of our all-atom simulation. So we have the WRF3A and M-dimer relaxed in the lipid. We also run a 41 M-dimers all-atom simulation in a flat lipid with the same composition as our envelope model. We put the M-dimers in random orientation so that it won't generate any artificial pattern. We observed this filament-like pattern forming after 3 microseconds and the clustering of M-dimer in the lipid bilayers after around 10 microseconds. We also compare the functional conservations between WRF3A and M-dimer. First, we annotate the positively, negatively charged residues as well as the polar residues in blue, red, and green respectively. We then annotated the functional residues of WRF3A here of the top cavity hydrophobic seal and hydrophilic groove of the ion channel, as well as the lipid binding and tetramerization structural sites in WRF3A. We then compare it with the M-dimer and find that the hydrophilic groove is not conserved between these two. However, we observed more charged residues in the center pole of the M-dimer, which means the water molecules and ions are passing through the transmembrane domain through the central pole instead of the hydrophilic groove as we observed in WRF3A. The lipid binding and tetramerization sites are well conserved between WRF3A and M-dimer. So we are expected to see similar behavior as we observed in the 41 M dimer all atom simulation shown here. We want to thank the TACC Supercomputer Center from University of Texas at Austin and the Julish Supercomputer Center to provide us the computational power we needed to complete our research. And here is our main reference. We have our envelope paper here and the two other reference on the M dimer as well as the OR3A structure paper. You can check our poster in the resource session, and you can see more details, which I cannot cover in this video. You're welcome to post any question you have, and I will try my best to answer them quickly. Thank you.